microorganisms are identified for practical purposes. For example, to determine appropriate treatment for infection. Most prokaryotic organisms don't have distinguished morphological features or even much variation in size and shape. Many different approaches are used in classifying and identifying microorganisms that have been isolated and grown in pure culture. For clarity, we divided them into two groups, classical and molecular. The most durable identification are those that are based on combination of approach. Classical approaches to taxonomy make use of morphological, physiological, biochemical, and ecological characteristics. These characteristics have been employed in microbial taxonomy for many years and form the basis for phynetic classification. When used in combination, they are quite useful in route identification and may provide phylogenetic information as well. Morphological features are important in microbial taxonomy. Morphology is easy to study and analyze, particularly in eukaryotic microorganisms and more complex bacteria and archaea. In addition, morphological comparisons are valuable because structural features depend on the expression of many genes and are usually genetically stable. Thus, morphological similarity often is a good indication of phylogenetic relatedness. The morphological features listed in this table are often employed in the classification and identification of microorganisms. Physiological and metabolic characteristics are directly related to the nature and activity of microbial enzymes and transport proteins. Because proteins are gene products, analysis of these characteristics provides an indirect comparison of microbial genomes. This table lists some of the most important properties. Enzymatic activities are widely used to differentiate bacteria. Even closely related bacteria can usually be separated into distinct species by subjecting them to biochemical tests. In this box, biochemical tests are used to identify bacteria in marine mammals. In the example shown in this figure, an unknown enteric bacterium is inoculated into a tube designed to perform 15 biochemical tests. After incubation, results in each compartment are recorded. Notice that each test is assigned a value. A code number is derived from scoring all the tests. A computerized interpretation of the simultaneous test results essential and is provided by the manufacturer. The ability of the microorganisms to colonize a specific environment is of taxonomic value. Some examples of taxonomically important ecology properties are life cycle patterns, the nature of symbiotic relationships, the ability to cause disease in the particular host, and habitat presences such as requirements for temperature, pH, oxygen, and osmotic concentration. Microorganisms have left almost no fossil record, so molecular analysis is the only feasible means of collecting a large and accurate data set that employs microbial evolution. Taxonomists can use an organism DNA-based composition to draw conclusions about relatedness. This base composition is usually expressed as the percentage of guanine plus cytosine. If there is a differential of more than 10% in their percentage of GC pairs, then these two organisms probably aren't related. Of course, Two organisms that have the same percentage of GC aren't necessarily closely related. Other supporting data are needed to draw conclusions about their phylogenetic relationship. The base composition of DNA can be determined in several ways. The most obvious is genome sequence. However, for microbes whose genomes have not yet been sequenced, the G plus C content often is determined from the melting temperature of DNA. If a double-stranded molecule of DNA is subjected to heat, the complementary strands will separate as the hydrogen bonds between the bases break. If the single strands are then cooled slowly, they will reunite to form a double-stranded molecules identical to the original double strand. When this technique is applied to separate DNA strands from two different organisms, it is possible to determine the extent of similarity between the base sequences of the two organisms. This method is known as nucleic acid hybridization. The procedure assumes that if two species are similar or related, a major portion of their nucleic acid sequences will also be similar. The procedure measured the ability of DNA strands from one organism to hybridize with the DNA strand of another organism. Hybridization of 70% or more indicates the two organisms belong to the same species. That's all. Thank you for your attention.